But today I'll be talking about the reactivity-guided design, spectroscopic monitoring, and scalable syntheses of novel antibody-drug conjugates. So kind of an introduction to the topic. Most modern cancer therapeutics involve chemotherapy, which is basically injecting drugs into your body in the hope that the drugs will be toxic to the cancer cells and remove those cancer cells from your body. However, this does have several significant side effects that aren't good for your body, including mainly killing healthy cells, which aren't cancerous. And this can cause hair loss, which you see in many chemotherapy patients, and also causes many autoimmune disorders and can risk sepsis. And also, this isn't particularly time effective. The chemotherapy may not affect the cancer for many weeks or months. And so this is where the idea of an antibody-drug conjugate comes into play. And an antibody-drug conjugate, or ADC, consists primarily of a monoclonal antibody, which targets an antigen at the cancer cell surface. So these antibodies are designed to respond to the antigens that are present on cancer cells and on cancer cells only, which leaves the healthy cells intact. And this also contains a release mechanism as well as a cytotoxic payload. And so this idea was first developed by Niels Jern, Georges J. F. Kohler, and Cesar Milstein. And for their invention, they won the Nobel Prize in 1984. And the idea was that once monoclonal antibodies were produced through a process involving antigen-specific plasma cells, they could be mass expressed in hybridoma cells. And these antibodies could then be used to connect to cytotoxic payloads and thereby produce specificity and selectivity in cancer therapeutics. And so since antibodies are such massive molecules, when talking about ADCs, it's kind of important to note the scale of the molecules we're working with. So as you can see on the right, aspirin is considered a small molecule of sorts, and compared to a monoclonal antibody, it's tiny. And so monoclonal antibodies are part of a class of molecules called biologics, which involve classes of proteins and other large molecules that are found in your body. And so this is just sort of a side note regarding the scale of the two types of molecules we'll be working with. And so, like I mentioned previously, an antibody consists primarily of the an ADC consists primarily of the antibody monoclonal antibody, which is designed to target cancer cells, as well as a linker and a cytotoxic drug. So the antibody's main purpose is to deliver the drug exactly to cancer cells, and the linker's job is to degrade under specific conditions and release the cytotoxic payload. So this is the mechanism through which an ADC works. First of all, the antibody will bind to the cell through the antigens that are present on the surface of the cell. And then the ADC will enter the cell through a process called endocytosis, through which the antibodies internalize into the cell. Within the cell, there will be certain conditions that will cleave the linker, thereby releasing the cytotoxic payload and causing DNA damage, which prevents the cell from replicating and causes cell death. And so ADCs are still an emerging field. As of today, there's been approximately 11 ADCs that have been FDA approved, and there's many more ADCs that are currently in different stages of clinical trials or FDA approval. So it's still an emerging field with lots of new innovations yet to come. And so when we were considering this concept, we came up with a pretty simple one-step synthesis for first-generation linker as a sort of proof of concept regarding our technology. So in this case, we used a molecule called 6-methyl-hexanoic acid and coupled it to SN38. SN38 is a derivative of camptothecin, which comes from a plant in China. And this plant has been used for many years for herbal medicine and things like that. So scientists look for derivatives of it, including SN38, which could be much more potent and cytotoxic. And so what we did was we coupled this 6-methyl-hexanoic acid, which has this linker functional group which has this molamide functional group on the left, which is the pentagon shape you can see. And that molamide functional group has been known to react specifically with sulfide bonds on the antibody, which allows for easy conjugation to the antibody. And so, as you can see, we were able to scale up our first generation SN38 linker to over 300 milligrams, and we conjugated over 15 milligrams to pertuzumab, which is an FDA-approved ADC. And we actually tested that in cells. And in this case, we also tested it out with another drug, MMAE, or monomethyl auristatin E. And so there's also different variations of MMAE, including MMAF and other variations, and we use the same linker, again, as a proof of concept. However, the difference is this linker wasn't designed to be cleavable through a controlled process. Rather, we were just testing its degradation through hydrolysis and other natural processes, without too much focus on like controlling it through an enzyme or other variations. And so one important step in the conjugation of ADCs is the reduction of disulfides on monoclonal antibodies. So disulfides are essentially two sulfur atoms bonded together, and they're present on cysteines, which are one of the amino acids that will make up a monoclonal antibody. And so what we do is we use a molecule called tris-2-carboxyethylphosphine, or TSUP, to break this disulfide into two sulfhydryls, or two individual SH bonds. And these SH bonds can then be attached to by the molamide. However, one problem with this process is that TSUP is designed to break these bonds, and leaving it in for too long with the antibody can actually cause the antibody to degrade, which you can see in this SDS page right here. You can see that after the first hour, the heavy chain of the antibody starts to degrade, and you see the light chain fall off as well. And so this is previously reported by Terence Kantner and other scientists, but 4-benzoic acid contains this azide group, which is these three nitrogens with different charges, and those can react with the TSUP to quench it, essentially preventing it from further reducing bonds. And so we use 4-benzoic acid to quench the TSUP and prevent degradation of our antibody. And one more metric that we use to characterize our ADCs is something called the DAR, or drug to antibody ratio. So what the DAR essentially does is it gives you an average of how many drugs are attached to each antibody. So this UV-Vis spectra that you see on the left has three main peaks: the peak at 220 on the far left, the peak at 280 on the black line in the middle, and the 380 nanometer peak at the blue line. The 380 nanometer peak represents the chromophore on SN38, our drug, and 280 and 220 are absorbance spectra caused by tryptophans and tyrosines in our antibody. And so what we can do is, by adjusting for molar mass and comparing the relative absorbances of these two peaks, we can actually determine on average how many drugs are attached to a single antibody. And this UV-Vis in particular has a DAR of approximately 8. So while our first generation had several pros, including the fact that it was easy to synthesize as it was a one-step synthesis, it does have one major flaw, and that is that this ester functional group circled in red is a hydrolytic liability, which means that in aqueous conditions, this linker will degrade pretty fast. And this is not good because mice bodies and human bodies are made up a majority with water, and so the drug falling off the linker before it was meant to fall off would be a major problem, as we would lose all the selectivity provided by the monoclonal antibody. And so we actually saw this happen experimentally. So right as we were gearing up for an in vivo study, our final conjugation before we we're going to inject into mice, our DAR following that conjugation was approximately 1.5. And so this was pretty bad considering we couldn't replicate the previous DAR of 8 to 9. And based on this UV-Vis spectrum, you can see that the SN38 peak is almost non-existent. And so we determined that the molamide had been attached, but the SN38 had fallen off. And so this was a pretty major setback for us, as it set us back like quite a few months and cost us a paper and possibly a grant proposal. But in order to rectify this issue, we decided to try something using palladium to remove the molamide. So in this case, people have reported that you can use palladium chloride to cleave a molamide off of a sulfide. And so we decided to test this out on glutathione and then progress to an antibody. Glutathione is a good substitute because it has this SH that you can see on the right. And this SH is more sterically accessible, meaning it's not encumbered by the complexity of an antibody. And so we were hoping to test out our removal process on glutathione before progressing to IgG, and that work is still in progress. In addition to testing out the removal in order to save the monoclonal antibody, which had the molamide stuck but not the drug, we also began work on our second generation linker. So the first generation linker was esterase cleavable. At that functional group that I circled in red would have been attacked by an esterase enzyme. But in this case, we took advantage of the fact that cancer cells have more proteases expressed by orders of magnitude over normal cells. And so we decided to try and attempt a linker that would cleave through the use of a protease. So in this case, this NH that you can see on our final product can be cleaved by a protease and will eliminate the drug, in this case MME. And it undergoes the same process for SN38. And so this is sort of the mechanism of action through which the protease will cleave our linker. So initially the linker will enter your cell through endocytosis, in which case it will enter a protease. The protease will attack this amide bond and go through what's called a 2,6-methide elimination, which releases this phenyl ring and releases the payload intact. And this finally causes apoptosis or cell death. And in order to synthesize this linker, one of the first reactions involved what's called an amidation, or the coupling of a carboxylic acid and an amide. So these groups circled in red, you can see the two amide groups, the two NHs, and the two carboxylic acids, the COOH and this functional group on the bottom. And previous literature reported protocols were giving us trouble with yield, as we only had 30% and 50% respectively. So we decided to figure out whether we could optimize these conditions for a more scalable synthesis. So we decided to first screen a variety of coupling agents, including HOT2, HBTU, TB2, EDC, and more. And these coupling agents fell into two major categories, which were carbodiimides and uranium salts. So what you can see on the right is these uranium salts have one thing in common, which is these three nitrogens and this benzene ring right here. And so that's what we call a benzothiazole, and that's not present on these carbodiimides on the left. So what we do is we use a molecule called HOBT to catalyze this reaction, and the HOBT is basically this benzotriazole with a hydroxy, and so that's how we compare relative yields and catalyze the reaction when we don't have the benzotriazole present. And so we quantified these yields through QNMR using 19F benchtop NMR because we used a molecule which had a fluoride on it, which allowed us to track the kinetics of the reaction. And so what we could see with 4-fluoroaniline was that HC2 actually provided us with the best yield, followed by PIBOP, and then HBT and TBT were pretty similar. And for 4-fluoro and methylaniline, in which the amine is secondary amine, meaning it has another molecule bonded to it and it's not an NH2, is that HA2 is actually the best coupling agent. And so using these relative comparisons, we were able to determine that we should either use HA2 or HC2 for our amine couplings. And then another part of our project involves the fluorescent monitoring of ADCs. And so what we do is instead of a drug, we can attach a fluorophore, in this case, rhodamine B or FTSE. And fluorophores are useful because, like I showed with the UV vis earlier, they'll absorb light at specific wavelengths and provide strong signatures for either attachment or detachment. And so we can quickly quantify how many fluorophores are attached and thereby monitor the process of bioconjugation. And so one thing you'll see is that both of these fluorophores have a similar polyaromatic ring system on the bottom, as well as another phenyl ring on top. The main difference is that FTSE has this SCN functional group or isothiocyanate, meanwhile rhodamine has this carboxylic acid. And so initially we tried to attach rhodamine B, firstly by converting it to rhodamine B ethylene diamine. And so we attach this amine to the carboxylic acid, which forms this new ring system in the middle right here. And then this conjugation process was pretty simple. We attached the 6-1-beta-hexanoic acid again as our first linker, and then we attached it to a monoclonal antibody, bovine IgG in this case, as a test substrate using the same conjugation process. And so what we saw, actually our results from this experiment were actually pretty unexpected. So again, we see the antibody peak at 280 nanometers and 220 nanometers, but we also saw this shouldering, or like the splitting of the peak. And we also saw a lack of absorbance at 550 nanometers, which wasn't something we expected because rhodamine is known to absorb pretty strongly at 550 nanometers. And so one thing we did notice was that the formation of the new product created this new ring system. And so we hypothesized that breaking this middle ring and forming a new ring with the added nitrogen would actually cause a loss of fluorescence. And so we decided to try FTSE instead because the isothiocyanate can be functionalized into an amine without changing the polyaromatic ring system, which allows us to still track the fluorescence and absorption of the fluorophore. And so another experiment we tried with rhodamine was attaching the rhodamine onto a linker that's already been conjugated to an antibody. So our current antibody conjugation process involves forming the linker and drug or other payload conjugate first, and then attaching the new molecule onto an antibody. But we had an idea. We thought, what if we put the linker on first and then we attach the fluorophore or drug or whatever? So we attached the 6-1-beta-hexanoic acid to the antibody, and then through another amine coupling with the NH2 and the carboxylic acid using EDC, we attempted to conjugate the rhodamine onto the antibody. And again, we saw a minimal absorbance by UV vis, but we're hoping that using FTSE instead will allow us to track this better, as we can still quantify the absorbance through the modification of FTSE. And then finally, one major component of this process, this is not an internal project. It involves collaboration with Sunny Bay Biotech. And Sunny Bay Biotech provided us with these proprietary antibodies that they've engineered to target triple-negative breast cancer, HER2-positive cancer, and other types of cancer. And their method for producing these antibodies involves injecting mice with the sequence of their antibody. These mice then generate the antibody in different types of cells, such as hybridomas. These antibodies are then screened and then humanized. And one major problem with this is that it's super costly. It costs thousands of dollars and even like hundreds of mice only to produce like 10 to 20 milligrams of antibody. So we also wanted to optimize this process so we could create more antibody and could proceed with conjugation at our own pace. So the final effort that we're currently considering is using transfection of plasmids to mass produce antibodies. And we're currently testing this with Herceptin, which is another FDA approved antibody. And basically what we do is the plasmids for these are commercially available and they contain the sequence for Herceptin. And then you can transfect the plasmid or basically inject it into CHO cells. And CHO cells will then reproduce the antibody sequence for you. And then you can extract the antibody from the CHO cells, and then you end up with often hundreds of milligrams or even a gram of antibody, which is much more than we'd ever need for an in vitro study. And so this is cheaper, quicker, and it's higher yielding. So this is what we feel is a beneficial path for us to consider. And then finally, acknowledgements. I'd like to thank Edward for helping us and advising us in this project, and then also all of our group members in ASDRP. And these are some photos of people who helped me with this project. So yeah, thank you.
Great job, Adam. That was fantastic. We'll take questions for our presenter today.
I want to give you extra credit for finishing right on time with 30 slides. I can't believe you're from Edward's team. Like, now you have become like interdisciplinary between Edward's team and perhaps my team, but I want to give you credit for that and thank you. What feedback I have is just like, if we can have like, divide your slide into first introduction and then what method you used, because a lot of people coming from not very strong chemistry background, if you make it more like educational, that they'd have a takeaway from this, it will be wonderful. Or just like in the middle of that, if you just want to bring everyone's attention that do they understand chemistry, like what drug conjugate was that? What element we have added? Can anyone write it in the chat? So just kind of engaging people who at some point feel like, oh, that's going to be beyond my knowledge. Okay. But beside that, amazing work. Well done.
Thank you. I'll definitely keep that in mind in the future.
Okay, I'm just going to ask my question. So, so you were saying they were using mice to get the antibodies, to create the antibodies. I'm wondering, why didn't they just take them from humans? Like, I'm sure we have things in our bodies that produce them, and we probably have a lot of things in our bodies that produce antibodies. So why don't we just like take just like how we like harvest bone marrow?
Yeah, so the antibodies are actually like engineered to be better than current human antibodies, so they definitely have a basis in like humanized antibodies. But the thing is, mice are just like much easier to extract and purify the antibody from, because this comes from the stomach and like you end up killing the mice in the end. So yeah, I don't think there's really a method from which we could like safely and scalably extract antibodies from humans at a rate which would enable like the mass development of these type of antibodies. So it's a possibility, but mice are just like more efficient, safer, and like it's just much easier to mass produce small, like the quantities of antibodies that are produced through mice rather than
Got
humans.
it. Thank you.
Other questions? Okay, so there's no other questions. Atman, great job. That was fantastic.
Thanks, Ed. Lorelai's laughing in the background right now. So
What's up?
Lorelai's laughing in the background.
Ah, okay.
It's off.
Yeah, certainly a lot of great science. And what to me, what's more exciting than just the fact that we're doing great science is the fact that we have such dedicated high school students who stay until 3 o'clock in the morning doing experiments. That's, quite crazy. So kudos to you, but also kudos to your parents for putting up with us.